Yogah Karmasu Kaushalam.
Good morning to all of you. I am Dr. Jigna Desai, Associate Professor from Department of Biosciences, Veer South Gujarat University. I know you people are from the different background of biological subject. Today we are going to talk something about the zebrafish as the animal model. Let's start our lecture. I am going to share PPT with you people. So this is the table of my content. First of all, we'll talk about something about the introductory aspect of zebrafish and zebrafish, why zebrafish is beneficial, maintenance of zebrafish, creating gene-based model in zebrafish, and zebrafish toxicology. As far as taxonomical aspect is concerned, the zebrafish is related or it is included into the Cypriniformis order, and the scientific name of zebrafish is Deniorario. In this image, you can see the zebrafish is having white and black strip-like pigmentation. So that's why particularly it is known as zebrafish. Now, general features, it is 4 centimeter, approximately 4 centimeter long, and sexual dimorphism is there. It is freshwater fish and tropical fish. It is omnivorous, and the female spawn every 2 to 3 days, and the reproductive aspect of breeding season will be throughout the year. So there is no particular breeding season except it being affected by the temperature and the light sometime. So the beneficial aspect of this fish is what? It is the universally available and low cost in maintenance. This slide shows the life cycle of the zebrafish. The zebrafish is having larval form also, and it is the larval form is transparent. Here you can see after fertilization, the 4 cell stage and 8 cell stage, the cleavage will start, and after that within 4 to 6 hours it will become the shear stage, and gastrulation and epiboly we can visualize under the microscope. After that the organogenesis will start and hatching will be there. After hatching, the larval form will arise, and this larval form will be having the heartbeat as well as the eye formation, and the somite will be there, and the larval form is pre-swimming and it will become adult after the juvenile form. So it is having total lifespan of 42 to 66 months, and it is good model for developmental studies because of the transparency and at its early stage. So here in this photograph you can see the fertilized egg of the zebrafish, and it is totally transparent, and you can clearly visualize the 4 cell stage and 8 cell stage also. And of course it is easy to handle and optically transparent because of its optically transparency, easy to study the embryonic development, epiboly, gastrulation and segmentation. The growth delay can be also visualized, and malformation or morphological abnormalities can be also visualized. It is easy to manipulate messenger RNA, DNA and fluorescent tag can be easily done. So micro injection can be also done very easily, but of course we have to visualize for the micro injection, we have to visualize this egg under the microscope, and the specific specialized expertise should be there to handle the ova, fertilized egg. These are the various developmental stages of embryonic life of zebrafish. Here you can see the 4 cell stage, 8 cell stage and high stage which be there, then dome same structure is there, 30% epiboly, 50% epiboly. Each and every stage we can clearly visualize under the microscope. So here you can see in this stage the 15 somites are visible. Here are the 17 somites are visible. So morphological deformity can be visualized very easily under the microscope in case of development of the zebrafish, because the larval stage and the zygote's embryonic life is totally transparent. Now why we have to utilize, or we can utilize very easily, why the zebrafish is being recommended vastly throughout the world? Because traditionally we were using the mice as well as the non-human primate for the biomedical research related with the human, but Their use is costly and studies at early developmental stage raise ethical concern also, because we have to think so many aspects related with the ethical concern whenever we are dealing with the primates and whenever we are dealing with the any other kind of mammals. Because in most countries the adoption of the 3 R's, that is replacement, reduction and refinement for animal research is mandatory and encourage the use of alternative model. For that purpose uh, we are shifting towards the animal cell line, and we are studying all the type of medical aspect in vitro human cell model. But it can impart the substantial information, but the model cannot be replicated by multi-system interaction observed in the human disease or in vivo animal model. Because in case of cell line research, the cell line are monolayer and single type of cells are available in the flask. So cell signaling aspect which is available in the three-dimensional portion of the system or the organ which is present in vivo, which is not available in vitro condition. So particularly we cannot correlate directly our outcome with the three-dimensional animal model if we are dealing with the human cell line in vitro for the biomedical research work. So conversely, invertebrate like fruit fly and Drosophila melanogaster and nematode can be also we can utilize. But we are using also for the many years. But obvious drawback of this invertebrate is that they lack similar brain structure and organ system to human, because they are totally different phyla and they don't have similar system level organization and same system and organ like humans, so they may not model for human disease accurately. So we are shifting towards the zebrafish. So why we can utilize the zebrafish and what are the advantages of zebrafish when we are using the zebrafish? Because as we have seen that it is having very short lifespan and short reproductive cycle also. So short reproductive cycle and the reproductive developmental embryonic development is totally transparent, so we can visualize all the type of development very easily. So short reproductive cycle is there. It is easy to maintenance in the large number in small laboratory space, because it is only 3 to 4 centimeter in length. So a very small structure of a small aquarium can be utilized, or we can utilize for the maintenance of the zebrafish, and in the 10 by 10 room also in the stacking type of aquarium system we can deal with the number of type of experiment, and we can maintain the number of zebrafish also. So zebrafish genome is fully sequenced. Over 70% of the annotated human gene have true ortholog in zebrafish genome. So it is very helpful to know something about the genetic aspect of the disease related with the human being. Zebrafish embryos are transparent, allowing non-invasive and in vivo imaging of the heart as well as the another system also. Simple genetic manipulation, for example, transgenic biosensor for different signaling pathways are available, can be studied very easily. Adult zebrafish are able to regenerate different organs. They can regenerate tail also, they can regenerate the heart also. So regenerative aspect of the heart, regenerative aspect of the tail or any kind of organ system we can study very easily in case of zebrafish, because it is easy to handle and observe also. The high throughput drug screening, that is low volume of drug, can be utilized for the study purpose because it is very small animal. The high number of offspring is there. High number of offspring, fecundity is very high in case of zebrafish, and the female will lay egg every 2 to 3 days. So fecundity is very high and offspring number is also very high. And of course the fertilization is external and the development is also rapid. So lifespan is small, external fertilization and rapid development, and over 70% of the annotated human gene have true ortholog in the zebrafish. So that's why this animal model is advisable and it is very resourceful as far as the biomedical research related with the human is concerned. Now another advantage of using zebrafish is zebrafish have been used to study numerous human diseases including neuropsychiatric and neurodegenerative disease owing to similarities with human brain physiology and anatomy, because the human brain physiology and anatomy, the brain of zebrafish is also having a midbrain, forebrain and hindbrain. They are also having diencephalon like structure. So physiological aspect and the anatomical aspect of the zebrafish brain is also very much similar as to the human being. To the date, model have been developed for some of these diseases including amyotrophic lateral sclerosis and Huntington disease. The zebrafish brain is comparable with mammalian brain with fore, mid and hind brain. They are having diencephalon, telencephalon and primary neurotransmitter function is generally similar to the human being. So neurotransmitter function, that means we can say the physiological aspect of the zebrafish brain or the nervous system is very similar to that of the human. Like mammals, zebrafish possess the blood-brain barrier also, and permeability tests indicate that its physiological properties are conserved between zebrafish and human. In addition to this physiological benefit, zebrafish also exhibit sophisticated cognitive behavior such as learning and retaining association, and they manifest well-documented anxiety behavior also. One of the key advantages of zebrafish is their suitability for genetic manipulation, which has led to development of thousands of mutant, transgenic and otherwise genetically altered strain of the zebrafish for the study purpose of various diseases related with the humans. Genetically altered zebrafish featuring genotype or phenotype of interest can be produced with less effort than as compared to rodent and other animals or other mammals, as DNA and RNA can more easily be injected at the single cell stage due to external fertilization and development of the fish. Besides traditional reporters like green fluorescent protein, GFP, genetically encoded calcium indicator like GCAM, and the optogenic reported channel rhodopsin can easily be incorporated into the transgenic zebrafish line also. So that's why we can deal with the some of the disease related with the eye also and this other sensory organs which are there in this human being. So zebrafish is very beneficial as far as physiological aspect is concerned, as far as anatomical aspect is concerned, and genetic aspect concerned, because it is very much similar to that of the human being and its physiology and anatomy. Now we will see something about the maintenance of the zebrafish in the laboratory. Here I have shown only one aquarium in which the harvesting is going on for the eggs of the zebrafish. So setting up a breeding tank, it is there, and we have to provide particular environment for the zebrafish maintenance. They require continuously circulating environment, and continuous aeration should be there whenever we are maintaining the zebrafish. The pH level should be between 6.8 to 7.8 and temperature should be between 26 to 28 degrees Celsius. So this type of environment will be helpful for the zebrafish. As far as breeding and the egg laying is concerned, they are very particular regarding the light, because and the in the early morning light, whenever the rays or intensity of the light is particular, then only the female will lay the egg, and we have to separate out the male and female at the time of breeding. In one tank you can have one female and two male in the one tank, and after overnight the dark phase they will lay egg in the early morning. Here you can see how we can collect the blood from the very small 4 centimeter organism, that is zebrafish, the blood collection method. So we can collect blood from the cardiac ventricle incision also. We can collect blood from the gill vessel also incision also. We can collect blood from the caudal arteria vein also. So specific type of specific type of gauge will be there, and we can collect the blood from the from the zebrafish. Now here the oral gauge method are also shown in the images. This is the oral gauge method. This oral gauge can be inserted into the mouth of the zebrafish, and you can inject or you can give the oral doses of any kind of toxicant or drug, and drug administration can be done with the help of this oral gauge methodology. Now, blood collection method from the zebrafish, how we can collect blood and how we can separate out the blood cell from the blood. So here the image is shown that we have, for that purpose, we have to excise the tail of the zebrafish with the help of sterile scissor. And after that, we have to insert our zebrafish with excised tail into the perforated 0.5 ml tube, that is centrifuge tube, microcentrifuge tube. And this microcentrifuge tube, again, it will get inserted into 1.5 ml centrifuge tube. And this second centrifuge tube must be having the heparin, which is the anticoagulant. After that, this centrifuge tube will be inserted into the microfuge centrifuge, and it will be centrifuged for 5 minutes at 550 rpm. Once again, the same procedure, centrifugation will be done at 10,000 rpm for 5 minutes. At the end of the procedure, we will have the differentiated blood cell at the in the form of pellet, and the plasma will be separated out, and we can collect plasma as well as the blood cell of the zebrafish. Now, creating gene-based model in zebrafish. How we can create the gene-based model in zebrafish for the learning purpose of various type of human disease like Parkinson disease, then Huntington disease, and Alzheimer disease, and neuropathy, as well as the skeletal deformities related with the humans. Now, first of all, we'll talk about the forward genetic approach. Many of the gene-based model utilize genetically altered strain of zebrafish, which are created using several methods. One of the method is plasmid containing a gene or nucleotide sequence of interest is injected to the zebrafish egg, and DNA is eventually can be integrated into the genome of the zebrafish. However, this method is of having low rate of success, as only about 5% of resultant fish possess the desired genotype. The Kawakami in 2007 have reported it. A notable improvement used the toll-2 system, which depend on the identified toll-2 sequence, which are found throughout the zebrafish DNA. Now, what is toll-2 system? The toll-2 system consists of transposes messenger RNA and toll-2 transposon donor plasmid, and is introduced into the fertilized egg by microinjection. Microinjection can be done as we have shown in the previous slide. The genomic integration of the toll-2 constructs are generated in the germ lineage and transmitted to the offspring very efficiently. So, this toll-2 system can be utilized to introduce the foreign DNA or gene of our interest into the genome of the zebrafish. Injectant transposes messenger RNA cleave the DNA at the particular sequence, and plasmid contain a toll-2 construct with the desired new DNA insert into the resulting gap. The method is much more accurate, and 50 to 70% of the offspring inherit the inserted DNA. So the in the previous method, only the plasmid containing the gene of our nucleotide sequence, it is having only 5% of success ratio. But then if we are using toll-2 system, then the success ratio will become the 15 to 70%. Now, this is the diagrammatical representation of the toll-2 system, which is consist of transposes messenger RNA and a toll-2 transposon donor plasmid. In this figure, we are seeing that it is being introduced into the fertilized egg by microinjection. First of all, it will introduce into the fertilized egg by microinjection. Thus, the genomic integration of told to construct are generated into the germ lineage and transmitted to the offspring very efficiently. So here you can see the injected transposon messenger RNA, after that cleave the DNA, and gene of our interest along with the plasmid will be integrated into the germ lineage of the zebrafish. So, and in this methodology we will have 50 to 70% of the success ratio. Now in forward genetic approach, the different method of generated zebrafish model of human disorder have been explored over the last decade. Initially, number of large-scale forward genetic screen based on random mutagenesis with radiation, chemical or insertional mutagenesis. Mutagenesis revealed the zebrafish mutant affecting different aspects of embryonic development and biological process. The phenotype-driven approach was also applied to screen for gene involved in the various system development disease. Mapping of causative changes established some of these mutants as model for human skeletal disorder. For instance, a study by Gistelin et al. Several type 1 collagen zebrafish mutant previously discovered in the forward genetic screen were established as representative model for the brittle bone disorder osteogenesis imperfecta. Now reverse genetic, another approach, that is reverse genetic approach, the Morpholino knockdown and gene editing. Now in this case, the reverse genetic targeting induced local lesion in genome which is known as stealing method. In that case, particularly the adult zebrafish male are being treated with ethyl nitrosuria, that is ENU, and mated with the wild type of female, that is the F0 first generation, F0 generation. The resulting F1 male progeny containing approximately 100 mutation in each germ cell. Now resulting F1 progeny, all the male will be having approximately 100 mutation in each germ cell and are grown to the breeding age and sacrificed to create the mutagenesis library. What is mutagenesis library? Mutagenesis library is consisting of genomic DNA used to screen for F1 male harboring mutation in gene of interest and cryopreserved sperm from each individual male fish for recovering the mutant line using the in vitro fertilization. So all the male from the F1 generation will be utilized for the preparation of mutagenesis library, because these male are having genomic DNA which are harboring the mutation in gene of our interest. And of course, from this male only, the sperm from this male only, they will be cryopreserved for the future aspect. In future, through the in vitro fertilization, we can recover this particular mutant line using this cryopreserved sperm. So in this procedure, result 50% are heterozygous mutant animal in the F2 generation. In F2 generation, we will have 50% of heterozygous animal. Heterozygous animal. The F2 carries the in cross to generate the F3 generation, and this F3 generation in which 25% of the offspring are homozygous for the mutation. So F2 generation will be having heterozygous, but this heterozygous F2 generation which carries the F2 carries are in cross to generate the F3 generation, and this F3 generation will be having homozygous, and they are homozygous in the mutation. This approach identified two zebrafish TP53 mutant line which can be utilized for the development for the study purpose and for the development of zebrafish to develop the malignant peripheral nerve sheath tumor, that is MPNST. We can study this nerve sheath tumor with the help of this modified or mutant strain of zebrafish, that is TP53. So this is the reverse genetic approach for the development of the mutant or the new mutant strain will be helpful for the study aspect. Now another methodology is zinc finger nucleases, that is ZFNs. The customized zinc finger nucleases can introduce target frame shift mutation with high efficiency and thereby enabling directed creation of zebrafish gene mutation. Here we can see the ZFN target site is having a left half side also and right half side also, and between the left half side and right half side there is a spacer which is made up of 5 to 6 base pair. Each half side contain 9 base pair sequence that can be recognized by the three finger zinc finger array. This half side, right half side and left half side can be recognized by three finger zinc finger array. This ZFN consists of zinc finger array fused to a nucleus domain. A heterodimeric pair of ZFNs bind to the left and right half side. They will bind to the left and right half side and induce a double stranded DNA breakup. This heterodimeric nucleus domain will combine with the double stranded DNA and they will create the break in the DNA break in the spacer. In the spacer space, they will create the breakup. Cell utilize non-homologous and joining machinery to repair the DBS, an error-prone process that can lead to random insertion and deletion at the target site. Whenever the breakup will be there, breakage of the double-stranded DNA molecule will be there, the cell will after that they will utilize the non-homologous end joining, that is NHEG, that is the particular repair mechanism of the cell itself, and after that it will be possible to do random insertion or deletion which is known as indel at the target site. So we can insert or we can delete a gene of our interest from this spacer site of the double-stranded DNA molecule. Now here is the graphical flow chart which is indicating the process of zinc finger nucleases, that is ZFNs customized zinc finger nucleases methodology. This particular methodology will have at least about 42 to 55 steps, and it will take more than 1 to 2 weeks. So after 1 to 2 weeks we will have one mutant or you can say genetically modified strain of the zebrafish which can be utilized for the further study of particular disease. Now the transient gene knockdown using Morpholino oligonucleotide which is known as MOS. MOS work by either blocking translation of target messenger RNA or preventing splicing of pre-messenger RNA. This method revolutionized gene manipulation as it was cost-effective and relatively easy to use. With many of studies reviewed here employed this method. It well documented that MOS may inhibit off-target gene in addition to the instead of target gene also. Sometimes the different gene will be targeted. In this case, in this type of Morpholino oligonucleotide gene knockdown system. So sometimes it is furthermore it is difficult to inject precise and reproducible volume of MOS into zebrafish egg. So there must be particular volume injection should be injected into the zebrafish egg. So it is very difficult to inject precise and reproducible volume of MOS in the egg. So this methodology is little bit difficult to exhibit, and of course sometimes it target the other gene also which are not of our interest. So that's why it is difficult. Another methodology is CRISPR-Cas9. It is known as clustered regularly interspaced short palindromic repeats, which is known as CRISPR-Cas9. The newest method, this is the newest method, which was developed from the bacterial immune defense mechanism. Emmanuel and Donna won the Nobel Prize in 2020 in chemistry for their work on the CRISPR. it is rapidly become very most widely used genetic editing technique. Why it is so? Because generally and specifically in the zebrafish, the first step is the construction of short piece of synthetic RNA. In this methodology, we have to synthesize, we have to construct the short piece of synthetic RNA that target the complementary segment of desired DNA molecule. The Cas9 protein, an enzyme that cleaves the DNA, use the synthetic RNA to identify the site of the cleavage. So this synthetic RNA will combine with the Cas9 protein, and they will form a complex, and this is known as what? CRISPR-Cas9 complex, right? It can be used. Now this CRISPR-Cas9 complex, they can identify the particular target site on the DNA molecule, and they will combine, this complex will combine with the target sequence, and it will cleave. The DNA will be cleaving. It can be used to excise gene or introduce a new gene also. So in both the way it can be utilized. We can insert the new DNA molecule, or we can delete the DNA molecule of our interest also, gene sequence also. CRISPR-Cas9 is cheaper and much less time consuming than the ZFN and TALEN. In zebrafish, CRISPR-Cas9 is highly effective. The mutation can be induced in 86% of fish on average and be completely inheritable. It is completely inheritable also. So it is very cheap, less time consuming, and more than 86% of the offspring will inherit the particular gene of our interest. So it is very much advisable, and nowadays it is widely used genetic editing technique, generally and specifically in case of zebrafish manipulation. This technique show immense promise for the creation of new zebrafish line. Now the generating of zebrafish transgenesis, combining the ability to create the custom transgenic line with the current imaging technique present a unique opportunity to study the function of genes in the living animal. Something not easily achieved with the other model. With advanced microscopy technique, imaging live transgenic line is becoming more accessible. Light sheet microscopy enable the user to illuminate and imagine the entire plane of tissue. Here we can see in the image, here we can see the angiogenesis in case of zebrafish, and here the developmental aspect of the zebrafish is there. The different stages and different tissues have been combined with the fluorescent dye, and the imaging system, that is fluorescent lifetime imaging microscopy. Through this type of microscopy system, we can visualize each and every developmental aspect, each and every system in this in its developmental phase. So it can provide the additional data on overlapping emission spectra and the intensity of the fluorophore also. So this particular type of imaging system can be utilized for each and every type of system during the development of the zebrafish. For example, development of the liver, development of the intestine, development of the eye, also development of the brain, and development of the heart also. So this type of imaging system is very much helpful for zebrafish transgenesis and the study. Now zebrafish in case of toxicological aspect, so ecotoxicology is there, environmental toxicology is there. So we can utilize, as we have seen in the previous slide, that zebrafish embryos are totally transparent, and we can visualize each and every developmental aspect, the embryonic life under the microscope. Right now, under my supervision, under my guidance, one of my student is working on the toxicological effect on the embryonic life, toxicological effect of delta methrin, as well as another compound which is bifenthrin. So they are using so deltamethrin is a pesticide which is being utilized for the agricultural purpose, and in our country and other rest of the country also the deltamethrin and bifenthrin is also being utilized in the very much amount. So the toxicological effect all deltamethrin and other compound is being visualized. The preliminary work is going on, the pilot study is going on. So these are the photograph have been taken in our laboratory. That is 4.5 milligram per liter of deltamethrin. After exposure of 48 hours, the embryo will show the spinal column breakup. After 72 hours, it will show the spinal column breakup. Here you can see the total breakup of the spinal column is there, right? So yolk sac extension and breakup will be also there after 72 hours. So day 9 will be there. So the yolk sac breakup is there. Here is the yolk sac, and here we can see the breakup of the yolk sac is there, and extension is also there. Normally the hatching of the zebrafish will be there within 24 hours. So yolk alteration, because of the yolk alternation, the edema will be there, and pericardial edema here you can see, and the late hatching is there. In the after 48 hour, in the day 3, the lordosis you can visualize. Lordosis means what? The uncontrolled, or you can say lordosis, that is the abnormal condition of the developmental aspect in the spinal cord. The zebrafish is the vertebrate animal, and we can visualize very clearly the developmental aspect of this vertebral column also. So lordosis, here you can see the curvature of the body of the larva indicate the lordosis in the 1 milligram per liter concentration, and exposure is 48 hours. Here are the 2 milligram per liter in 48 hours. The kyphosis is there. Kyphosis is there. Spinal column breakup once again, and the lordosis, reduced pigmentation will be also are also observed. Pigmentation can be seen very clearly in case of larval stage of the zebrafish. So here you can see in other larval stage, here you can see the clear cut of pigmentations are visible, but as the concentration of the deltamethrin is increasing and the exposure time is increasing, the larva become less pigmented, and the reduced pigmentation will be continue after 42 hours and 72 hours. Some of the larvae have been seen without the pigmentation also. 0.5 milligram bifenthrin is we have observed the interaction or the reaction toxicity of bifenthrin in various concentration, that is 0.5 milligram per liter and 1 milligram per liter. Here you can see in 0.5 milligram per liter the breakup of the spinal column is observed after 72 hours, and after 96 hours the lordosis have been observed. Lordosis is there after 96 hours. The pericardial edema, yolk edema is there. Here you can see pericardial edema, we can clearly see. Of course, whoever is observing the zebrafish regularly, they can only understand what is the edema and what is pericardial edema. So we have to we should know the anatomical aspect of the zebrafish larva. Then only we can clearly visualize, or we can interpret our result when whether it is morphological deformities related with the somites, related with the pigmentation, or related with the skeletal system, or any other system is there, or neurodegenerative system is there. In 1 milligram per liter concentration in 24 hours, yolk alteration is there. Here you can see the yolk is reduced in case of the embryonic part. This is the unhatched embryo, and yolk inside of the embryo is altered. Edema is also have been observed. In 72 hours the scoliosis is there. Here you can see the lordosis is there, and here the scoliosis is there. Scoliosis, lordosis means what? The curvature of the vertebral column will be upward, and in scoliosis the curvature of the vertebral column will be rightward. So here in the curvature of the vertebral column is on the right side. So the it represents the scoliosis after 72 hours of 1 milligram per liter concentration of the bifenthrin. After 96 hours, the kyphosis was observed. Kyphosis means what? The it will affect the anterior portion of the embryo, as well as it will affect the pigmentation part, as well as the breakup of the vertebral column will be also there. Now the conclusion, the zebrafish become a powerful model organism for developmental studies, genetic research, drug and toxicology screening, and for understanding tissues regeneration and repair. In contrast to other vertebrate models such as mice and fertilization occur externally, which together with transparency and rapid embryo to larval transition permit easy access to visualization of development. Moreover, due to its rapid growth, a recognizable and complete vertebrate body plan is already in place by 1 day post fertilization, and embryogenesis is complete by 3 day post fertilization. In contrast to other vertebrate models such as rodent, the small size and large number of offspring of zebrafish allow for increased sample numbers, thereby increasing the statistical power of the experiments. Finally, the relatively low husbandry cost further contribute to the increasing popularity of the zebrafish as a model for research. These are some references. Thank you very much for listening me. Thank you very much, Dr. Jhala, coordinator of the program, for inviting me as a resource person and giving me opportunity to deliver my talk on this zebrafish, a resourceful animal model. Thank you very much.